Deep inside of each of our bodies, inside the organs, inside of the tissues, inside of the cells, lies a tiny nucleus, and inside of the nucleus sits DNA, the genetic material for most of the living organisms on Earth. Amazingly, this durable code is made up of only 4 different kinds of building blocks: A, C, G, and T. Certain parts of this DNA, called genes, act as templates for the production of proteins. Other sections of DNA are used to help turn genes on or off. There are as many as 25,000 genes scattered throughout human DNA. Intriguingly, many of these genes are also found in other organisms, with an almost exact copy of many human genes found in mice and other living things. One example is a gene called ZIC4. This gene helps control other genes. In mice, this gene is important for brain development. The ZIC4 gene is found in a wide variety of organisms, including humans, chimpanzees, mice, and even zebrafish. Why does this gene occur in all these organisms? The answer is that the gene was present in a common ancestor of all the organisms. Over millions of years, the genes were passed down from generation to generation in different lineages. Within each lineage, mutations randomly occurred in DNA, but many of these changes were lost over time. The changes that remain in a lineage are called substitutions. Because many genes are found in common ancestors, we can compare the sequences found in different species. These comparisons help us explore how the DNA that makes up the gene has changed or stayed the same over time. Let's compare one small stretch of DNA, just 20 nucleotides, from a control region near the ZIC4 gene in the human and the chimpanzee. What do you notice? There are no nucleotides that differ between the two regions. We can also compare the human DNA to the DNA from the mouse. Here you see one substitution. Now we'll compare the human gene to the gene from a zebrafish. Would you expect more substitutions compared to the human and mouse comparison? The comparison shows that there were 3 substitutions. We can also compare 3 genes to each other. Let's explore a comparison of the human gene, the mouse gene, and the fish gene. This can start to get difficult to decipher as we add more and more species to our comparisons. Another way we can show the same information is with a diagram called an EvoPrint. To make an EvoPrint, we pick one organism to be the reference sequence. Let's choose the human sequence as the reference. Now we'll compare it to the chimpanzee sequence. As before, there are no differences. Now we'll add in the sequence from the mouse. The EvoPrint shows substitutions as lowercase letters with a different background color. See how the changes show up in the EvoPrint? But just as importantly, notice how many of the letters have not changed. They have been conserved through natural selection. Finally, we'll add the sequence from the zebrafish. A quick look at the EvoPrint shows us which nucleotides have remained the same over time and those that have changed. To help us understand EvoPrints in even more detail, let's think about time. Let's start with a comparison of the human and chimpanzee gene. Remember, we're using the human gene as our point of reference. Humans and chimps last shared a common ancestor about 8 million years ago. This means that for 8 million years in the human lineage and 8 million years in the chimpanzee lineage, there were no changes in this section of DNA between these species. That's a total of 16 million years. Now, consider a comparison of the human gene and the mouse gene. Humans and mice last shared a common ancestor about 90 million years ago. The human lineage has been evolving separately for 90 million years. The mouse lineage has also been evolving separately for 90 million years. Put together, that's a total of 180 million years. This means that it took at least 180 million years for the one substitution we see to take place. Substitutions we can detect in the DNA through the EvoPrint. What about an EvoPrint that has 3 species: humans, chimpanzees, and mice? To find the total time in this EvoPrint, we have to add up the time involved on each branch of the tree of these species. In other words, we have to add up the time in all the vertical lines on the tree. In this case, this gives us 90 million years for the human lineage, 90 million years for the mouse lineage, and 8 million years for the chimpanzee lineage. That's a total of 188 million years. We can keep the process going as we add more and more sequences. Let's add in the gene from the fish. Humans and fish last shared a common ancestor 455 million years ago. How much time is represented in this EvoPrint? We have to add up the time for each branch in the tree, just like before. In this case, we add 8 million plus 90 million plus 455 million for the lineage leading to fish and another 455 million for the lineage leading to humans. This gives a total of over 1 billion years. See how all the vertical branches were included? How can we use comparisons of DNA sequences to learn something about evolution? One way is to look for stretches of DNA that haven't changed over very long periods of time. Scientists called these conserved regions. There were mutations to these regions, but the changes did not persist. Conserved regions can shed light on basic functions shared by many organisms. This is an EvoPrint for a larger portion of the DNA near the ZIC4 gene that we examined before. If we compare this region of DNA for a wide range of animals, we see that many areas are conserved. The red highlighted region shows just one conserved region. In this section, there have been no changes that have remained, none. This section of DNA has remained unchanged for over 477 million years. Even when we add in the sequences for rat, platypus, chicken, lizard, and zebrafish, there are still regions that did not change. This EvoPrint covers over 2.1 billion years. These must be special sections of DNA to stay so similar over these many millions of years. We know that mutations randomly occur in DNA sequences in every individual, of every living organism, of every generation. And many sections of DNA, like the sequence shown on the top, accumulate lots of changes or substitutions. However, regions of DNA that remain the same in different organisms over a long period of time, like the conserved region on the bottom, deserve special attention. They demonstrate that natural selection has acted to conserve this sequence over time. The sequence is so key to the organism's survival that random mutations are weeded out of the population. This is called purifying selection. There are tens of thousands of conserved regions scattered throughout the human genome. Scientists are just beginning to identify these conserved regions of DNA and figure out their function. With access to the web, you too can discover regions in our genome that have remained unchanged for eons. Understanding the role of these evolutionarily hardened sequences is an important frontier in the quest to understand the complexities of life. Turn on your computer and go explore.